So, what if you could build a human embryo completely from scratch? No egg, no sperm. Well, that's not science fiction anymore. Scientists have actually done it using just stem cells, and it's something that could completely change how we think about the very beginning of life.
All right, let's just jump right in. Take a look at this. It looks, for all the world, like an 8-day-old human embryo. It's got all the right parts in all the right places. But here's the absolutely mind-blowing part. It was grown entirely in a lab. No egg, no sperm, just a really carefully designed set of instructions given to stem cells. This is a brand new window into the first few moments of human life.
But you might be wondering, why go through all this trouble in the first place? Well, it's because this model helps us finally peek inside what scientists have called the black box of our own development. You see, those first 2 weeks of life are just this period of unbelievably complex change. This is literally when the blueprint for human being is first laid down. It's also, tragically, when a huge number of pregnancies fail, and it's where many birth defects get their start. But we've known almost nothing about it, because studying it directly in humans, it's been impossible. And the main reason for this gap in our knowledge is a super important ethical guideline, the 14-day rule. For decades, this has been the international standard. It says you can't grow natural human embryos in a lab for more than 2 weeks. It's an absolutely essential safeguard, for sure, but it's also the very thing that created that scientific black box. So that leaves us with this massive question, right? How do we unlock the secrets of our earliest beginnings to hopefully prevent diseases and miscarriages without crossing those critical ethical lines? Well, this new research offers a really powerful answer. You create a model that has the architecture of an embryo, but you build it from the ground up, entirely from stem cells.
So how in the world did they do it? The best way to think about it is like a biological recipe, combining just the right ingredients in exactly the right way.
The main ingredient, the flower of this recipe, if you will, is pluripotent stem cells. These things are incredible. They're master cells that haven't decided what they want to be when they grow up. They have the potential to become a brain cell, a skin cell, or, in this case, all the different parts that make up an early embryo. So this recipe calls for 3 very specific things. First, you need what are called naive stem cells. These form the epiblast, which is the core structure that down the line becomes the fetus itself. Next, you use a special chemical cocktail to coax other stem cells into becoming the building blocks for the future placenta. The life support system. And finally, you use a different chemical cocktail to create cells for the yolk sac, which is like the embryo's first little pantry, providing all the early nourishment. And that third ingredient, that was the key. This chart shows a huge breakthrough. See, making those yolk sac cells was always the hardest part. The old ways were totally hit or miss. They only worked about 16% of the time. But this new RCL chemical mix the team developed, they totally nailed it, boosting the success rate to over 60%. This wasn't just a small improvement, it was the game changer that made the whole experiment work. Okay, so once you have your 3 ingredients, you just mix them together and you let them work their magic. And this is where it gets truly amazing. The whole process plays out over 8 days. On day 0, the 3 different cell types are mixed together in a very specific ratio. Over the next couple of days, they start to clump together. On day 3, they're moved to this special shaker that gently rolls them around, kind of mimicking the environment inside the womb. And by day 8, something absolutely incredible has happened. These simple little clusters of cells have, all on their own, organized themselves into this complex structure that looks just like a 14-day-old human embryo. This is the concept of self-organization in action, and it's just wild. The researchers aren't sitting there micromanaging every little step. Nope. They just provide the right starting materials and the right environment. And from there, the cells start talking to each other, following their own internal genetic blueprint to build this incredibly intricate structure. It's just a powerful reminder of the intelligence that's already encoded in our own cells.
But of course, the big question is it real? I mean, how accurate is this model? How do we know it's a faithful copy of the real thing?
Well, the visual evidence is just stunning. On the left, you've got a classic textbook diagram of a natural 14-day embryo. And on the right, that's the lab-grown model. Just look at it. You can clearly see the same key parts. There's the epiblast, that core part that will become the fetus. You can see the cavities for the yolk sac. You can see that outer layer of cells that would go on to form the placenta. The resemblance is just undeniable.
But it's not enough for it to just look right. It has to be functionally right, too. And this table shows that it is. Scientists can check for specific proteins that act kind of like name tags for different cells. So, for example, a cell that's part of the future fetus, it should be wearing an OX4 name tag. A yolk sac cell, it should have a SOX17 tag. And guess what? The model has all the right name tags in all the right places. This is the proof that it's not just some hollow lookalike, it has the right cells in the right spots doing the right job.
So this breakthrough doesn't just solve a scientific problem, it opens up a whole new world of possibilities, and with it, a whole new set of really important questions for all of us to think about. So why does all this matter? Well, it gives us a tool we've never had before to tackle some of the most heartbreaking problems in medicine. We can finally begin to study why so many pregnancies fail in those first few weeks. We can investigate the origins of genetic diseases right when they're starting. We can even test if a new drug is safe for pregnancy without ever using a natural embryo. It is a profoundly new platform for understanding and protecting human life.
Now, it's really important to keep all this in perspective. The science is revolutionary, absolutely, but the technology itself is still in its infancy. That number you see, less than half of 1%, that's the current success rate for creating one of these perfect models. So while it's an incredible proof of concept, it also shows us just how unbelievably complex and difficult this process still is.
At the end of the day, this technology gives us this extraordinary new power to peer into the very first moments of our own creation. It could answer questions that we have had for centuries. But as these models get better and better, as they become more and more like the real thing, it forces us to confront a whole new frontier of questions. What are the ethical boundaries now? And how do we, as a society, decide together what comes next?